everyone, welcome back to the cell signaling course. In previous class, we were studying GPCRs, particularly types of receptors, type of G-alpha proteins, and the type of signaling they continue further. If you recollect, we discussed about one of those G-alpha and the signaling downstream of that, which was about the G-alpha S, which stimulated the formation of cyclic AMP. Today, we will see more of different types of signaling which go downstream of G-alphas. And we will also see how certain regulations happen on G-alpha signaling and take examples of each of G-alpha functioning in the system. So, just to recapitulate, what we studied in the previous class was about the G-alpha S. S stands for stimulation of cyclic AMP production. So, here what we know is that many of the G-alpha S, they were also studied in context of olfaction. That is why they are also called G-alpha olf. And the basic idea here is that this G-alpha once comes in contact of the receptor, particularly when ligand has bound here, or what we call here agonist. When that has bound, a receptor comes in contact with this G-alpha beta gamma subunit, and this is where when the exchange of GTP happens and G-alpha becomes GTP bound, it dissociates from the beta gamma subunit, and this is now active. And one of the things which it activates is the enzyme which is called adenylyl cyclase, which was earlier inactive because it was bound to the regulatory subunit. So, cyclic AMP production allows it activates. So, once the GTP bound G-alpha is activated, it now works on the adenylyl cyclase, which is further activated to produce cyclic AMP like molecule, cyclic nucleotides. And basically that cyclic nucleotide is binding to this kinase, protein kinase A, which was kept inactive by virtue of binding of its regulatory subunits. Once cyclic AMP binds, the catalytic subunit is free, which activates pKa, that phosphorylates the CREB, cyclic AMP response element binding protein, and which allows the transcription of molecule inside the nucleus. So, in case of the process of olfaction, what we know that the nasal epithelium is where this thing is happening. A lot of GPCRs are involved in it, and they are responding to the odorants, different odorants of different small molecules. So, in the epithelium you have cells which are support cells, and there are specialized cells which have these projections on their surface, and these projections have these receptors embedded in their membrane, and this is where they bind the odorant. If I say odorant receptors, they will bind to the odorant, and what will happen as a response to this binding inside, we know adenylyl cyclase is active, CAMP is produced. And what will eventually happen because of this odorant which has bound over there and cyclic AMP has produced, pKa is active, CREB is phosphorylated, transcription goes up. Downstream of CRE, cyclic AMP response element, this is where CREB will bind and activate transcription. And this is good enough to send the signal, because in response to some of these transcription and pKa activity, the channels will be opening which are present over here, and they will depolarize this. The membrane, which now sends the signal through this olfactory nerve. To the brain region. So, binding of odorant activates the GPCR, which activates this process and PK. PK now activates some of these channels which are present on the membrane of this neuronal cell, which depolarizes the membrane, and this depolarization is an electrical signal which is transmitted to the membrane, which is transmitted to the brain for perception of sensory olfactory cues. So, this was one of the first G alpha which we studied, G alpha S, and we also said this is the kind of class 1 or type 1 G alpha. The other G alpha, which is opposing or working opposite of what G alpha S was doing, this is what we call G alpha I, because this inhibits cyclic nucleotide. This can be done by inhibiting the adenylyl cyclase, or it can be done by activating an enzyme which is called phosphodiesterase. So, it can inhibit adenylyl cyclase or it can activate phosphodiesterase. What phosphodiesterase does? It takes cyclic AMP and converts into AMP, which is a linear molecule. This is good enough to ensure that the enzymatic activity of pKa are restricted, because adenylyl cyclase is not active. Where it works is wherever cyclic nucleotide gated things are working. So, here what we will see as example, particularly in case of our rod cells, where cyclic AMP gated channels are there, which are affected by the activity of G alpha T. So, normally what happens in this signaling is that ligand binding, as we saw, it activates the molecule like PDE, phosphodiesterase, and this will have its consequences. So, let us discuss this particular aspect in context of rod cells which are present in our retina. So, if you recollect one of the earlier classes when we were discussing type of ligand and the nature of ligand, this is where I suggested that the most abstract form of a signal or a ligand is light, where it is just a photon which induces changes. So, our eyes, particularly in retina, we have different type of cells which are called photoreceptor cells. We know that there are two photoreceptor type of cells, rod cell, which help us see things in low light, and the cone cells which help us perceive bright light as well as the color which is there in the light. So, let us think about this rod cells which are present over there. These rod cells have the GPCRs, and these GPCRs are bound with the compound which is called retinal. So, in normal state of these cells, when there is no bright light, retinal is bound to the GPCR, which is also in turn which is also seeing the production of cyclic GMP like molecule, and this sets up a situation where these rod cells are generating a current which is called dark current, because in the absence of light this current is being generated by the flow of ions across the membrane. So, there are these unique structures which are present here in the rod cell region, and this is where you will have the GPCRs enriched, which are bound to the retinal. So, what light does is that retinal which is bound to the GPCR is present in all cis situation. This is the position 11 and 12 of the carbon in the retinal. Both the hydrogens are in cis position. When light falls on it, visible light, it becomes transretinal. So this hydrogen flip happens and this is becoming a transretinal. This itself is good enough for activating the receptor. Now what happens because of that? So once the receptor is activated, now think about a situation like this where you have the rod cells, the segment which you saw outside. Here I told you that GPCRs are present, and the channels which are here, which are allowing the current, basically flow of sodium ion, which is normally happening, and the receptor is inactive. When light falls on it, the situation reverses because now cis retinal changes to all transretinal, which activates the receptor. The receptor activation meaning it will activate phosphodiesterase enzyme. What this is going to do is cyclic GMP which was there will be broken down into a linear GMP molecule. The channels which are here, these channels, they are ligand gated channels, which means that they bind to cyclic GMP and open those channels. So in the dark condition, cyclic GMP is more because there is no PDE activity at that point of time. Cyclic GMP binds to its channel, opens the channel, and flow of sodium ion happens. That is where dark current is produced. Now when the light has shone on these rod cells, all transretinal is generated, which activates the receptor. Cyclic GMP is converted to linear GMP by activity of phosphodiesterase, and now in the absence of cyclic GMP, the channels which are here, they are closed. So no current. And what has changed here? That GPCRs which were here, they are now active and PDE activity has gone up. So this is what is working over there. So if you are generating cyclic GMP in this situation, you should also have that cyclase enzyme. Since it is a guanine nucleotide situation, it is called guanylyl cyclase. So in this portion of your rod cell, you have the two enzymes, guanylyl cyclase and the phosphodiesterase, which are helping in the formation of cyclic nucleotide, cyclic GMP, and making it to the linear GMP. And this interconversion, because of the light shining and changing the retinal conformation and activating the receptor, is what helps visualize or have this dark current and help visualize things in dark, versus when you come out into from a dark room, if you come out, suddenly bright light happens, you cannot see anything because this conversion is going on. Now what happens in case of other G alpha, which is our G alpha Q? Here G alpha Q is the next G alpha type. You should remember all this while that G alpha is the one which has the GTPase property. So this G alpha Q, it has a unique mechanism where the ligand binding is not dependent on adenylyl cyclase. Rather, it is about the activation of another molecule which is called PLC beta. If you remember, we had discussed in context of receptor tyrosine kinases, where we saw phospholipase C gamma unit was working, binding to the receptor and breaking down PIP2. So the function of the PLC remains same, isoform changes here. So what will happen that when ligand is absent, you would not see any activation of the pathway. When ligand bounds here, it will activate the receptor, same G alpha will go into the GTP bound state, and this will activate this PLC beta. What PLC beta will do? It will recognize the PIP2, which is part of the membrane, break it down into diacylglycerol and IP3. So the two molecules will be generated from PIP2. Now this beta gamma unit which has been separated, they also work in this process and helps in the activation of protein kinase C. So how the system goes forward, that two entities which are being generated from the PIP2, they basically have independent function. Like IP3, it binds to the IP3 receptor which are present on the smooth ER surfaces, and smooth ER surfaces are considered as the store for calcium ion. Once IP3 binds to its receptor on smooth ER, it opens up the channel and influx of calcium happens. When I say influx, which is telling that more and more calcium is coming inside the cell, cytosol. Now over there, calcium mediated responses will be carried out, but one more thing which happens over there in this process of G alpha Q activity is that diacylglycerol is generated, which is a membrane entity, and this is required for activation of protein kinase C. In addition to diacylglycerol, protein kinase C also requires calcium for activation. So by G alpha Q signaling and generation of IP3, you have also released lot of calcium in the system, and diacylglycerol along with calcium activates protein kinase C to its full extent, and that's how it will downstream of itself, that's how downstream of its activation PKC will be phosphorylating multiple of those proteins as a effector molecules, and this protein kinase C is again a serine threonine kinase. So we will see how this moves forward, but also remember that there is a regulation on these lipids which we saw in case of receptor tyrosine kinases also, where we saw example of PI3 kinase, how they were working in an antagonistic manner, and if you recollect, they were class 2, class 1B of these PI3 kinase which had the kinase activity. We discussed that how this class is going to show its work with the GPCRs. Class A worked with the receptor tyrosine kinases, but these kinases do not work alone, they need an adapter molecule. So in case of receptor tyrosine kinase, we saw it was P85, P55 and P50 kind of adapter. Here it is P101 type, and this together binds with the beta gamma subunit and regulates the signaling. So remember what PI3 kinase will do, it will take PIP2 as one of the substrate and convert it into the PIP3, and that is how it will work opposite of what PLC is doing. So PI3 kinase family also comes into picture. However, this is not one of the directly activated molecules downstream of G alpha. In this case, the directly activated molecule is PLC beta, but the product which is getting generated or the subunits of G heterotrimeric G protein beta gamma are engaging into this conversion by PI3 kinase and thus having an opposing function in the signaling. Let's take an example of this signaling which goes on in most of our taste sensation. Whatever our taste buds help us assess, whether it is sour, sweet, bitter and so on, is because of this GPCR signaling which goes on in the cells which are part of taste buds. So this is shown, this particular image shows you a kind of taste bud where you have multiple of these cells which receive input from these cues, gustatory cues, and then activate the G alpha of G alpha Q type. So what will be happening? This is your receptor GPCR which will bind to the taste entity and it will activate G alpha Q. We know that receptors have that Jeff activity which will activate the G alpha Q, and this will activate PLC. This PLC will break down PIP2 into IP3 and DAG. IP3 will increase the calcium. In addition to that, there will be calcium which will be moving inside these cells. So this image which you see here is representation of one of these cells. So calcium moves in, sodium moves in, and there is a depolarization which happens. It is very well established fact that calcium is required for the process of exocytosis. So these taste bud cells have neurotransmitter filled vesicles. When you have more of calcium influx, whether from extracellular space or from internal stores, they help facilitate the fusion of neurotransmitter vesicles to the membrane and release neurotransmitters. And what is happening to this released neurotransmitter? There are these axonal ends which is shown here. They will be receiving these neurotransmitter and conducting the signal in form of action potential moving forward. So basically again exocytosis is getting converted into electrical impulse as a action potential and reaching to the brain for further analysis. So this is what something which we are seeing for the G alpha Q. We already saw G alpha S and G alpha I. What exactly goes on in some of the studies which has been shown in greater detail when it comes to ligand binding? If you recollect, I discussed in previous class that how the 7 helices are arranged so that G alpha can be engaged here for productive signaling. A small rearrangement in these helices which are part of the 7 transmembrane spanning regions. These G alpha binding regions of different helices are reoriented so that they can engage the G alpha, thus producing a small change in conformation. So some of the studies which are reported here, they show that particularly the alpha helix 6, that undergoes changes. If I say transmembrane alpha helix 6, that undergoes some changes and which allows it to be an inactive state or active state of the receptor. Consider a situation where this is the orientation of helix 6. This is the normal situation, and if I say intermediate position 1. So this is the position under normal circumstance when receptor is inactive. The other helices will be around, remember that, but this alpha helix 6 which is being concerned here. So when ligand binding happens, it actually pushes on this alpha helix and this helix moves to different conformations. Basically it can move to this position or it can move to this position. So more proper of a ligand, it will push the helix to the maximum displacement position. If consider this is your position S3 and this is position S2. So ligand binding is moving this alpha helix from one position to the other position, and because of this movement there is a space which is created here. Now in the ligand binding active state, the alpha helix 6 is oriented like this. Other structures, they kind of remain same. This generates a space for G alpha to bind. So normally the space which was not available, when ligand binds, it moves the alpha helix 6 to a different location. That is how G alpha can bind and activate the receptor. This happens in case of G alpha S, which is involved in cyclic AMP generation, as well as to the other G alpha, which is G alpha T family. Remember G alpha T for transducin, which was involved in our visual perception, which is also called G alpha I. So something similar happens here, is that there is an orientation of the alpha helix 6 like this. A retinal is bound here, other helices are present, but when light is shown on this and it converts from 11 cis retinal to all transretinal, there is a change in this position of the helix and it moves further to this position. What it does, it actually gives space around the other helices, and now G alpha T can bind. So what you are noticing here is that in the previous class what we saw, that helix movement is something which was activating the receptor, and this is what we are seeing, that what exactly goes on out of those 7 helices, that helix 6 actually is gaining more prominence and it is inducing a change, conformational change in the receptor, which opens up the space for G alpha to come and bind over there. What will happen over here when G alpha has bound, or in this case where G alpha has come and bound? Remember, the receptor has the Jeff activity, right? Jeff activity is part of the receptor. Now what this Jeff activity will do, it will convert this from a diphosphate bound situation to G alpha T or S into triphosphate bound situation, which is active. And we saw in case of S, it was working through cyclic AMP production, increase adenylyl cyclase activity, but here it will work through phosphodiesterase and reduce cyclase activity. So what we have understood in the class today is that the active G protein, which is in the class 1, activates adenylyl cyclase, which participates in the sense of olfaction, and it works through cyclic AMP molecule, increase availability. The active G alpha proteins of class 2, they inhibit the adenylyl cyclase activity, or they activate phosphodiesterase, thus decreasing the level of cyclic nucleotide. The sense of visual perception, that involves this kind of signaling where the PDE activity is increased and cyclic nucleotide levels go down. The active G alpha protein of the class 3, which is G alpha Q, it activates PLC. This PLC breaks down PIP2, releases calcium from interstellar store and activates the protein kinase C through DAG, diacylglycerol, and cooperative function from the calcium. And the taste of sense of taste or the gustation is something where we are seeing G alpha Q coming into picture. Thank you.